Good day and welcome to another lecture series on biochemical reasoning. My name is Iberemo. Don't forget to subscribe and click on the notification button for more videos on biochemical reasoning. This is the second lecture, and today we'll be looking at different questions with respect to biochemical reasoning. Well, I told my student that this course is available for year 2 students, year 3 students, year 4 students, 500 level, or even MSc or PhD students. So as far as you're a student of biochemistry, you must be able to answer some questions that are related to biochemistry. You must be able to reason biochemistry. So the first question we'll be looking at here, first question we'll be looking at is, why is protein degradation important for cellular function? So why does the cell need to degrade this protein? Of course, we know that these proteins have a definite lifespan. And when they've reached their lifespan, it is necessary for the body to degrade them. Now, this degradation can come as a result of a misfolded protein or damaged protein. So if the protein is damaged, there is need to degrade that protein so that the protein doesn't cause further damage to the cell. If the protein is misfolded, of course, we have seen the cases of misfolded protein like the Alzheimer's disease, the Parkinson disease, and the cystic fibrosis. So this is as a result of protein misfold. So when there is this misfold, the body needs to degrade those proteins so that the body can eliminate the protein, so that the protein doesn't cause damage to the cell. So let's take a look at the little answer I actually placed here. Proteins have a definite or a finite lifespan and need to be degraded or replaced when damaged. Misfolded or no longer required protein by the cell. Degradation pathway like the ubiquitin, of course, we know that when there is a tag of ubiquitin to a protein, I mean that protein is destined for destruction or for degradation. So we look at the ubiquitin protein system. This system targets and breaks down specific proteins. This turnover eliminates dysfunctional proteins, prevent protein accumulation, and allows for rapid adaptation to cellular needs and environmental conditions. So, for example, if you're fasting, at that point you're not taking any food, so those proteins that are necessary for digestion will not necessarily be synthesized because you don't need them. So the body will look at, instead of which is breaking this protein for this proteins that are required for maybe food digestion. So because of you're not eating, because of your fasting, and there is no food in your gut, so there will be no need to produce those proteins. Rather, the protein will not be broken down. The ones that are already available will not be broken down into amino acids that can be used to produce glucose or ketone bodies. But the ones that are used to produce glucose, those amino acids are called glucogenic amino acids. The ones that are used to produce ketone bodies are called ketogenic amino acids. So that when they call them glucogenic amino acid, means that their carbon skeletons can actually be used to produce glucose. While the ketogenic one, their carbon skeletons of the amino acid can be used to produce ketone bodies. So due to cellular needs and environmental conditions, the cell will determine which protein to produce and which protein not to produce. So second question, you have two cells undergoing aerobic respiration and the other undergoing, one undergoing aerobic respiration and the other undergoing anaerobic respiration. In which cell will glycolysis produce more ATP and why? So you have two cells. One is aerobic glycolysis, one is running under aerobic glycolysis, and the other one is running under anaerobic glycolysis. So the question is in which cell will glycolysis produce more ATP and why? Of course, glycolysis produce more ATP in aerobic respiration. That means in the cell undergoing aerobic respiration. Because in aerobic respiration, the glucose will break down into pyruvate, yielding two molecules of ATP. Now that pyruvate is further decarboxylated by pyruvate dehydrogenase complex to acetyl-CoA. The acetyl-CoA enters the TCA cycle, producing NAD plus NADH, about 3 NADH. Think about one FADH2 and one GTP. The 3 NADH further undergoes enters the electron transport chain for subsequent production of ATP. So for aerobic glycolysis, more ATP is produced, usually 36 molecules of ATP. Why under anaerobic respiration, the cell produce approximately 2 to 4 molecules of ATP. Because in that case, instead of the pyruvate to undergo the decarbonylation to go into TCS cycle, it will be converted to lactate via the enzyme lactate dehydrogenase. So that conversion will regenerate NAD plus that is necessary for glycolysis to keep running. So at that point, that is the branch point where it is the cell would need to regenerate the NAD plus for continuation or maybe for continual glycolysis. So let's look at the other question. Okay, see, in aerobic cells, okay, the aerobic cell produce more ATP. Glycolysis directly generates two ATP molecules, but in aerobic respiration, the pyruvate enters the citric acid cycle and oxidative phosphorylase, which I just explained, generating up to 36 molecules of ATP per glucose. So while the anaerobic respiration relies on glycolysis and fermentation, which only yields 2 to 4 ATP molecules per glucose. So you can see that 2 to 4 molecule is as a result of conversion of pyruvate to lactate with subsequent production of NAD plus that goes back to glycolysis, usually the step 6 or the first oxidative state of glycolysis, where conversion of glycerol 3 phosphate to 1,3-bisphosphoglycerase for glycolysis to continue. So we have another question here. So how can analysis of protein sequence reveal evolutionary relationship between different organisms? We know that the analysis of the protein sequence gives us a relationship between proteins or between two organisms. For example, if you take the sequence of hemoglobin in man and the sequence of hemoglobin in fish or in dog or in cow, you'll see that the hemoglobin have similar sequence. And that similar sequence tells us that they function similarly because the hemoglobin in man is actually for transportation of oxygen. The hemoglobin in dog, in fish, in cow is for transportation of oxygen. So even though the amino acids are not exactly the same, they have a similar sequence. And with that similar sequence, they have similar function. So it can actually make us to predict the evolutionary relationship between organisms. It can also help us to predict the mutation or where there is a difference between two organisms. So let's look at the little question the little answer I put in place here. So comparing protein sequence from different species can provide insight into their evolutionary history. Similar sequence suggests a common ancestor, while difference can indicate mutation or adaptation that have occurred over time. This information helps us to understand the relationship between organisms and the emergence of new function from existing proteins and the process of biological adaptation. Now, in biotechnology or in genetic engineering, we look at the phylogenetic tree. You can actually plot the phylogenetic tree of different organisms, and that will actually show us the ancestors of where that particular sequence must have originated from. For example, we might be interested in looking at the type of Salmonella typhi that causes typhoid within our vicinity. Now, if we look at if we are able to isolate the different, if we are able to isolate the typhoid, the organism that causes typhoid, which is Salmonella typhi, within the Owerri metropolis, we also isolate some of this same this thing within the environment of Lagos, within the environment of Port Harcourt, and we look at why is it that the salmonella typhi in Lagos is more resistant to a particular drug comparing to the salmonella typhi present in Port Harcourt or present in Owerri. So we can actually use the sequence of their gene makeup. We can actually look at the sequence of their gene makeup and trace where there is a difference between them. So if you look at if there is a mutation between the genes from the ones in Lagos, the salmonella typhi in Lagos, the salmonella typhi in Port Harcourt, salmonella typhi in Owerri, and we sequence their gene. So if there is any mutation in the gene, we can actually predict the resistance to that particular drug must have been due to the mutation in this particular gene on this particular protein and the rest. So that will give us an insight of where there is mutation and where there is evolutionary ancestors, or if they have a common ancestors. So now there's another question we have here. Say, during intense exercise, your muscles switch from aerobic respiration to anaerobic respiration. Explain how this switch helps maintain ATP production even in the absence of sufficient oxygen. Now, what they mean here is that there is oxygen, but the oxygen is not sufficient. So what the muscle does, the muscle will have to switch to anaerobic glycolysis in order to maintain glucose supply. Now, the body also look at the Cori cycle. In the Cori cycle, we look at in the muscle, the glucose is converted to lactate, right? While lactate leaves the muscle into the bloodstream and be transported to the liver. In the liver, the lactate is converted to glucose via gluconeogenesis. After the conversion to glucose, the liver pushes the glucose out. The glucose finds their way back to the muscle for continuous energy supply. So that is actually called the Cori cycle. There is also alanine, glucose alanine cycle, where the pyruvate that is produced is converted to alanine via alanine aminotransferase. So when they convert it to alanine, the alanine enters the bloodstream, down passes, is transported to the liver, where the liver will convert the same alanine back to pyruvate using the same enzyme, alanine aminotransferase. So the enzyme will convert alanine back to pyruvate. The pyruvate will be converted to azol acetate. From there, we go back to gluconeogenesis, where glucose is produced. The glucose produced leaves the liver back to the muscle for continuous supply of glucose, so that the muscle can continue with the exercise. Now, that is how the muscle can maintain ATP production. The pyruvate that is like the previous one I explained, the pyruvate that is formed in anaerobic glycolysis and in the presence of or the absence of sufficient oxygen is converted to lactate. To generate that conversion, we use lactate dehydrogenase, while NAD plus is regenerated that continuous glycolysis. So that continuation ensures that ATP is available for continuous muscle contraction and relaxation. So we'll be ending this lecture 2 with this question. So if you have not subscribed to this channel, please do so to subscribe, and do not forget to click on the notification buttons. Click on like and make your comments, so where we can actually have an improvement in this lecture. Just make your comment, ask your questions, and we'll attend to them in the comment section. So the answer is glycolysis is regulated by several enzymes, including phosphofructokinase, high concentration of ATP and citrate, along with low ADP and AMP levels. Now, this ADP and AMP level inhibit the PFK1 with the phosphofructokinase 1, slowing glycolysis, allowing however, during exercise, ATP consumption increases and oxygen supply might be limited. This increase in ATP and increase in AMP stimulates PFK1 activity, leading to an increased glycolytic rate and faster generation of ATP through fermentation, compensating for the temporary lack of oxygen. Thank you very much. We'll see you in the next lecture, Biochemical Reasoning 3. Thank you very much.